Hey everyone, welcome to 6.3 Defense Against Disease
The immune system.
video.
Here is
This is
a
all
specification if you want to read it.
pause it now and have a quick. Okay, so we're going to start with the first lines of defense. The first lines of defense are the things that stop pathogens from actually entering and infecting our bodies in the first place. There's quite a few first lines of defense, but for IB, you don't actually need to know a huge amount of them. So you need to know that the skin is a physical barrier that stops pathogens from entering the body. Obviously, when the skin is injured, pathogens can then enter. And mucus is found in a number of places in the body. And what the mucus does is it can trap harmful particles and stop them from actually getting into the body. So, for example, mucus in kind of the nose and throat stops any particles from the air, or it stops a lot of particles from the air and catches them. And then we can get rid of them out of the nose and mouth or by swallowing them. Okay, so if the skin is injured and a blood vessel is broken, then blood clotting has to occur in order to stem the flow of blood and ensure that you don't lose too much blood. Now, so the first thing that happens is platelets are released from the platelets release clotting factors. Sorry, I thought I'd got that wrong there. So the platelets within the blood release clotting factors, and there's a number of clotting factors, but you don't need to know about them specifically. The clotting factors cause prothrombin to be converted into thrombin. So prothrombin is an inactive molecule, whereas thrombin is active because the thrombin then causes fibrinogen to be converted into fibrin. The fibrin then forms a sort of mesh over the injured blood vessel, and it stops the blood cells from leaving, or stops the blood from being able to get out. Fibrinogen is soluble, but fibrin is insoluble, and that's obviously useful because you don't want blood clot to dissolve within the blood, because then it wouldn't be doing its job. So we need to know this little cascade here. It comes up in exam questions quite often, actually. And then finally, for this slide, we've got some stuff about coronary thrombosis. Now, coronary thrombosis is a blood clot in a coronary artery. A thrombosis is a blood clot in at any point, whereas coronary thrombosis specifically in the coronary arteries of the heart. And what happens is the blood clot, the thrombus, blocks the artery, which means that there's a lack of blood flow to an area of the heart. And if there's a lack of blood flow, there's a lack of oxygen that gets there to that area of cardiac muscle, and this results in a heart attack. You also need to know a little bit about atherosclerosis. So atherosclerosis is where plaques form within the arteries, on the walls of the arteries. This can lead to thrombosis. Because within the plaques, the blood cells all can get caught and build up, and if that gets bigger and bigger, then it could block the artery, or it could break off and move to a smaller blood vessel and block that. Risk factors of atherosclerosis, you need to know these are smoking, hypertension, which is high blood pressure, high cholesterol, diabetes, obesity, and lack of exercise. Okay. So you also need to be prepared to analyze data related to coronary thrombosis and heart disease. There's loads of data out there on heart disease, so be aware that that could come up in a question. Okay, this flowchart that you're about to see is going to outline the entire immune system. Now, you'll realize quite quickly that if you Google the immune system, it is massive. There's so much to know. Now, everything on this slide is all you need to know. There's a couple of little bits from this slide that I've taken from HL, but I think that it means that you understand it better, and students always tend to struggle with the immune system. So here we go. So we've got the first lines of defense. So they stop the pathogens getting into the body in the first place. However, if the pathogens do get into the body, phagocytosis occurs. Now, in phagocytosis, you can see you've got your little bacterium here. The phagocyte, which is this blue cell here, it's a type of white blood cell, ingests the pathogen. So it engulfs it by endocytosis, and the bacterium moves into this vesicle here, which is called a phagosome. Now, that then fuses with a lysosome. Now, you've learned about lysosomes, they contain digestive enzymes. And the digestive enzymes, you can see them here, the blue spots, they are going to digest the pathogen. And then Within this phagolysosome, so this is the phagosome combined with the lysosome. Within the phagolysosome down here, you now have all of the broken down bits of the pathogen, and then we can get rid of them via exocytosis. Phagocytes are non-specific; they will just engulf anything. So they wouldn't find, for example, an HIV virus and be like, "Oh no, I don't want to ingest that." They will just ingest anything. Interestingly enough, sometimes phagocytes, the white blood cells, can even ingest sperm in a female's body because they're deemed to be foreign. So anything foreign could be ingested by phagocytes. What then happens is, instead of getting rid of all of the parts of the pathogen, they keep hold of some of the antigens, and then what happens is this phagocyte will present the antigen on its surface. So the phagocyte takes in the pathogen, breaks it down, gets rid of everything apart from the antigens. The antigens remain on the surface, and we then call it an antigen presenting cell. And essentially all it's doing is showing that antigen to the rest of the immune system. So it's kind of saying, hey guys, to all of the other white blood cells, hey guys, this is what you're looking for. Like, this is foreign. You need to find it. So it just kind of alerts the immune system to be able to better find the pathogens. After that occurs, the antigen presenting cell causes the T lymphocytes or T cells to start dividing. Now, T lymphocytes are another type of white blood cell. You might be realizing there's a lot of types of white blood cells. They then divide into either T killer cells. The T killer cells you don't need to know very much about. You could just know that they infect body cells and destroy them. That's enough. If you Google it, you'll find loads. T helper cells activate the division of B lymphocytes or B cells, which are another type of white blood cell. The first type of B cells are plasma cells. Oh, I should have not had these underlined. Sorry, guys. Plasma cell, now you need to know this, produces and secretes antibodies. This comes up in multiple choice questions, short answer questions all of the time. And memory cells, this is just HL, you don't really need to know this for SL, are cells that remain in the body for a long period of time and activate the rapid secondary response when a pathogen is encountered for the first time. Now, this here is a massive overview of the immune system. If you learn nothing else about the immune system, just learn this, because actually there's quite a lot of information there. Okay, HIV. So HIV is a viral disease. It's transmitted by bodily fluids such as blood, semen, vaginal fluid, and breast milk. You can't catch it any other way. It has to be transmitted by bodily fluids. So you wouldn't get it from touching, or you wouldn't get it from using the same towel as people, or kissing, or anything like that. So just via bodily fluids. The HIV virus decreases the number of T helper cells produced in the immune system. So we saw T helper cells on the previous slide. You can go back if you need to have a look. The T helper cells activate the B cells. So if we lower the amount of T helper cells, you're also lowering the amount of B cells. HIV results in sufferers contracting opportunistic infections. So opportunistic infections are infections that usually you wouldn't really show any symptoms for, and your body would just fight them off. However, because people with HIV, their immune system is lowered, then their immune system can't fight off these infections like it normally would, so they actually kind of take hold and produce symptoms. People with HIV are also more likely to develop certain cancers because obviously the immune system is weakened. And then lots of people don't really understand the difference between HIV and AIDS. So you can have the HIV virus, but you don't necessarily have AIDS. You only have AIDS when you have a collection of diseases as a result of the HIV infection. Now, every patient will have a different collection of diseases depending on which infections they've been exposed to, depending on their environment, or depending on the specific DNA that they have. So everyone who has HIV will get, if they develop AIDS, they will have a different AIDS. So AIDS doesn't have specific symptoms. It very much depends on the individual. Similarly, not everyone who has HIV will develop AIDS. There's loads of drugs that you can take now. HIV used to be a death sentence, but it very much isn't anymore if you're diagnosed and treated. Okay, on to antibiotics. So we should already know that antibiotics only kill bacteria and not viruses, because viruses are non-living and they don't have a metabolism. The reason that the antibiotics affect the bacteria and not the human cells when we take them is because the prokaryotes have, the bacteria have a specific DNA replication and specific cell walls that we don't have, and they affect them in that way. Bacteria become resistant to antibiotics, which means that they can't be killed by antibiotics. Do not say that they're immune to them, because that's not a thing. Also, people can't be resistant to antibiotics. So people could be immune to disease. Bacteria can be resistant to antibiotics. Some bacteria can become resistant to lots of antibiotics, and that's when it's a real issue, because if they're resistant to all antibiotics, it means that you can't treat them. You have to let the immune system do its job, or the person might die. And there's in fact, it also says here that they can become resistant by mutation or by sharing their plasmids. So what they do is they swap plasmids, and that means that both bacteria will be resistant. Steps that can be taken to combat that antibiotic resistance issue is that doctors don't prescribe antibiotics unless someone really needs them. People need to complete the course of antibiotics because the weaker bacteria are killed first. Good hygiene standards, and antibiotics not being used in animal feed. So farmers used to put it in animal feed to make sure that the animals didn't get sick and therefore didn't die, and they could sell them on and make money. Okay, in order to be able to upload, I'm going to stop here. So, and then I will do this slide by itself in a separate video. So it's a smaller video.